I think when we were discussing about this presentation, what is going to make, I told that we were not going to start here. So take us somewhere, different location, show us something. And when the food was being served, I was again the MHC mode. The one gut is handling so much different food, locating everywhere, and things are going nice. So show us the magic of that one molecule which handles almost all kind of various things, self or non-self presented, and how it works. Thank you. And see, you are getting standing ovation before even starting.
Thank you, Sanket. Good afternoon, everyone. I think we all had good energy boost and then a lot of heavy chain displacement into our brains. So let us move on with one more heavy topic, that is MHC. But I think after a good meal, we need a vacation, right? So come on, guess. What comes to your mind? Louder. Australia, fine, great. So in Australia, we often miss this small island. This is Tasmania, great. So Tasmania is famous for its beautiful beaches, wonderful mountains, and also the Tasmanian devil. Yeah, Tasmanian devil is a marsupial. It's a mammal, like a kangaroo, but a way smaller animal. In 96, what happened is a lot of these animals were there, but by 2009, almost 90 or almost 80% of them were killed, and the animal was declared as endangered species in Tasmania and Australia. Now they're slowly growing back. Yeah, why did they die? They died because of this tumor. This is the devil facial tumor disease. This is a cancer of the face. So why should a cancer kill 80% of the population? It usually does not happen that way, right? It's infection that kills. COVID came, it killed. Fine. But cancer should not kill like that. So for that, veterinarians and scientists started looking at their behavior and found that these devils tend to bite each other during mating, during fights, during territorial fights, all these things. So when they bite, they found that they seeded this cancer tissue into their face or body or wherever. So it caused this facial cancer disease and then spreading. Simple? All of you are convinced? Okay. If you're convinced, let us do some time travel. Let's go back to 1940. Peter Medawar was sitting with his wife Jane in Oxford. He was sitting in a gardening in the afternoon. And this was World War II time, and one Royal Air Force aircraft comes and crashes about 200 meters away from their house. After the crash, they go for help. They go to help the pilots, and they find these pilots severely burnt. So the ambulance comes by then, picks them, puts them into the burn ward, and luckily, because Peter Medawar was a zoologist, he had some experience dealing with tissue culture and cell culture, so he was called upon by surgeons treating these patients, burn patients, to help them in grafting, skin grafting. So he went there all excited to learn new things. They tried to graft, but they found that when they grafted from other people, the skin grafts failed. Now we all know this, but in 1940 it was a new thing. And when the graft was taken from the same person, maybe from the thigh or somewhere, it was accepted. And then when they did a biopsy from the failed graft, what they found is there was a lot of immune infiltration. Yeah. So this baffled Medawar. It should not happen, right? Skin is skin, it should have been taken, and why? From where is this immune cell coming all of a sudden? So he went back to his lab, did what he does best, took 25 rabbits, cared for them, fed them, cleaned them, everything. He did it all alone. He did series of complex experiments, transplanting skin from each of these rabbits to the other. That means every rabbit had got about 24 skin patches. So then he documented what happened to each of these skin patches very carefully. And they found that when the grafting was done between non-related rabbits, the skin graft died. Okay. And you take a bigger patch and put it died faster. But when they grafted from related rabbits, from their litter mates or from the same progeny, then it stayed. So coming back to time traveling again, coming back to 2009, where this facial tumor was, simply proposed to be spread by biting, it should not happen, right? A skin can't come, how can another tissue come? It should not. So why did not these seeded tumors get destroyed like Medaver's skin transplant experiments? This was a question to be answered in 2009. So they went ahead, did a biopsy of the facial tumor and found there was no immune infiltrate. Whereas Medaver's skin had a lot of immune infiltration upon rejection. And they further saw and they found that as compared to normal cells, there was this element that was not being expressed on the skin surfaces. That is, this is nothing but MHC. So MHC, if you can't remember anything from my talk, you can take home this message. MHC is a structure which can determine self from non-self, which can activate immune system, which is key in immune transplant, cancer, and also tolerance. Yeah? Are we clear with what MHC is? So we often get this confusion of what MHC, HLA, we keep using this again and again. It's very confusing. So let us clear it for once. So what is this? This is a cell, this is our DNA, it is coiled and made into a chromosome. Chromosome has got a short arm, long arm. We know all these things. So we also know that each arm of the chromosome is inherited from one of our parents, and they'll have alleles, other things. So MHC is the segment of the 6th chromosome, which lies in the short arm of the 6th chromosome. Yeah. So MHC is a glycoprotein that is expressed on the cell surface. To express something, we need a DNA sequence that actually translates to it, right? So MHC is the DNA sequence that is present on the 6th chromosome short arm. It is very small. It only accounts to 0.5% of the whole genes in the human genome. How is it inherited? The DNA itself, it is codominantly inherited. Codominant means one part is from each of our parents, and both are expressed. Usually what happens, if it is dominant, the recessive one won't be expressed. If it is recessive, both will have to be same and it will be expressed. Here, though both are different, they are both expressed. Yeah. Okay. So I promise I'm going to stop confusing you here. So we all have studied this again and again, and we have forgotten it again and again. So MHC is class 1, class 2, class 3. Yeah. Everything on chromosome 6. We are still in chromosome 6. We have not gone away from chromosome 6. So class 1 has got A, B, C. It also has got some non-classical MHCs like G, F, and E. Class 2 has got a lot of other things, DA, DP, DQ, along with DO and DM. We'll come to all those things later. And class 3 is mainly complements, TNF, and lymphotoxin, about which we are not going to discuss much today. So MHC as a gene, we know what it is. It's on chromosome 6. How do we name MHC? Yeah, all of us know HLA-B27, right? How is it named? So HLA is the HLA prefix, human leukocyte antigen. A is the gene, that is which class out of class 1, if it is A, B, or C, we just saw it. And then the star is there, which is a separator. So if it is starred, that means this molecule was detected by PCR or other molecular techniques. If it is not a star and just a hyphen, then it means that it is detected by other serological methods. And then after that, we have the 02, which is the allele group, like HLA-B27 now. Yeah, 27 will be our allele group. Then we have the specific HLA protein group, which comes after that. So it is up to here that is currently clinically relevant for us. So I'll give you an example again with HLA-B27, which is the protective HLA-B27, as in which HLA-B27 does not have spondyloarthritis. Louder, please. 06, right? It is HLA-B27 and here 06 at the level of HLA protein. This is where the difference is, right? Whereas if it was 04 or 05, then it would have been a pathological variant. So we know how to name HLA, we know what it is, where it is found in the DNA, but actual work happens on the cell surface. Yeah, this is another picture which we all see. So this DNA segment will code to some proteins, and these proteins are also called HLA, and they are also expressed on the cell surface. Yeah. So again, we'll only deal with class 1 and class 2. So the class 1, if you see, which is to your right, to your left. So the class 1 is made up of a heavy chain and a beta 2 microglobulin, which is not derived from chromosome 6. The beta 2 microglobulin is derived from chromosome 15 long arm. So heavy chain has got alpha 1, alpha 2, alpha 3 subvariants, and it has got a transmembrane domain and then a cytoplasmic tail. So a peptide which has to be bound to it binds to the groove between alpha 1 and alpha 2. So because they are two joined together, it is kind of covered in class 1. So because it is covered, it can allow smaller peptides to bind to it. Yeah. Whereas if you see class 2, it is open. It's kind of open, right? Two different heavy chains are there. Alpha 1, alpha 2 at one side, beta 1, beta 2 on the other side. And the peptide binding groove is between these two open heavy chains. So this allows us to present or to attach a longer peptide. So just to revise, where is this class 1 present? Class 1 is present on all nucleated cells, and class 2 is presented on majority of the antigen presenting cells, including the B cells. So the same thing again, yeah. So just looking at the difference, the polypeptide chains for HLA1 is alpha and beta 2. Alpha has got alpha 1, alpha 2, alpha 3. Class 2 MHC has got alpha 1, alpha 2, beta 1, beta 2. Yeah. So polymorphic residues means where is the majority of polymorphism happening? It happens at the peptide binding groove. So in MHC1, it should be at alpha 1, alpha 2. In MHC2, it should be alpha 1 and beta 1, between alpha 1 and beta 1. So again, as we told, the size of peptide that is binding is smaller for MHC1 because the groove is closed. It's not open. So 8 to 11 amino acid residues, whereas for MHC class 2, it is about 10 to 30 amino acid residues. Yeah. So we are done with the DNA part. We are done with what is expressed by the DNA. Now coming to the most confusing part, how does it express what it expresses? So for that, let us just get back to our central dogma, what we already know. We know how a protein is synthesized. DNA transcribes to RNA, RNA translates to protein. Any doubts with this? No, right? All of us know this. So here, if we see, this is the same thing, which is put in more detail actually. So from DNA, the mRNA comes, attaches to the ribosomes. Ribosomes will attach amino acids, cause a peptide. In peptides will undergo folding. What causes the folding is a series of other proteins called chaperones. Yeah. If you can't remember other chaperone names, just remember chaperones. That's enough. So chaperones will cause folding. Once the peptides are folded, becomes tertiary quaternary structure, they are loaded onto the Golgi complex and pushed out of the cell, either to the surface or into the lysozyme. So this is how any protein is synthesized in the body and expressed. So when it comes to MHC, it is no different, right? So MHC1, from the central dogma understanding, if you see, there are a lot of peptides that are translated, and then these translated peptides, because they are variable in size, they have to be chopped. What is going to chop? Because we have seen MHC1 can only take up to 10 peptides, and whereas MHC2 can take up to 30 peptides. So what chops them? These are proteosomes. But peptides are not just coming from inside the cell. Sometimes they can come from outside also. So when they come from outside, other proteases take over based on the number of amino acids available and the type of amino acid. They are also chopped. And if you come to this figure, once they are chopped by proteasome, the protein enters the proteasome chops it into smaller amino acids and peptide chains. The peptide chain will try to enter the endoplasmic reticulum through a complex called TAP. Yeah. So once it enters the TAP, that is where MHC class 1 is sitting. MHC class 1 is already sitting in rough endoplasmic reticulum. So now there is a new peptide that has been transported into the rough endoplasmic reticulum using the TAP molecule, and that is where the loading takes place. Yeah. So MHC1, yeah, let us go to how it is assembled. So I told you to remember chaperones only, right? What is its function? Protein folding, great. So MHC1 and alpha chain is sitting in endoplasmic reticulum. Then there are these chaperones, ERP57 and calnexin. So when they combine, they bring beta 2 microglobulin towards MHC class 1. So now it becomes a complete MHC class 1 assembly. So once beta 2 microglobulin attaches, the calnexin is released, and now a new complex including tapacin and calreticulin will work to bring this complex close to the TAP complex. So together this is called the peptide loading complex, because the peptide has entered through the TAP and now it has to be loaded onto the MHC. So it is called peptide loading complex. No creativity involved at all. Sometimes what happens, the peptides are longer than expected, so then they are again trimmed by the ERAP, endoplasmic reticulum aminopeptidase, that we already know of, ERAP1, ERAP2, all these things, polymorphisms. So ERAP is sitting inside rough endoplasmic reticulum, it will chop these peptides and then load it onto the MHC, that is MHC heavy chain and beta 2 microglobulin complex, and then all these chaperones are released and MHC class 1 molecule gets into the Golgi apparatus and is transported outside the cell. So what type of peptides are MHC1 representing outside? Those that are inside the cell or outside the cell? Inside the cell. So this is called endogenous peptides. So MHC1 will present endogenous peptides to which cell? CD8 cells. Yeah. So now moving on to the next MHC, that is MHC class 2. We already discussed, this is present on antigen presenting cells mainly. So which are our antigen presenting cells? Monocytes, dendritic cells, sometimes B cells. Yeah, these are our antigen presenting cells. It's a little different than MHC1 because here also the MHC2 complex is sitting inside the endoplasmic reticulum. But then it is constantly cycled within the lysozymes so that it comes in contact with the peptide that comes from outside. So here, because antigen presenting cells will take peptides from outside, right? It will take up an antigen that is outside, that is exogenous, and then bring it in and then process. So that other things don't come and bind to it, MHC is, the peptide binding groove is covered by something called invariant chain. Yeah. So with this invariant chain, MHC class 2 is cycled all around the cell. You can see it better here. So MHC class 2, once the invariant chain comes and attaches to the peptide binding groove, it goes everywhere. It gets into the lysozymes, it gets into the phagosomes, and again comes into the MHC2 binding complex, which is the lysosome. Yeah. Here, the externally acquired amino acids or peptides are taken inside, degraded in the lysozymes because of changing pH, and then it is brought to the MHC2 complex, MHC2 loading complex, where with the help of HLA-DM, another non-classical MHC, the invariant light chain, which has been degraded into a small molecule called CLIP, that is then removed by HLA-DM. So sometimes what happens is this is not needed, so HLA-DO is there, which actually stops HLA-DM from doing this work. So HLA-DM removes the CLIP. Once the CLIP is removed, the antigen binding or the peptide binding complex is open, and then the externally acquired peptide can come and sit there. Once it sits, again the same pathway, it is transported into the Golgi complex, and then these cells go out. Yeah, let us just look at it again. So I told you already, MHC2 is sitting inside the rough endoplasmic reticulum. There is an invariant chain there which prevents any non-specific binding. Then it is taken through the Golgi complex into the lysozymes. In the lysozymes, the invariant chain is partly degraded. Only the CLIP is remaining in the MHC2 peptide binding site. Here, when it gets the exogenously acquired antigen, which is processed by endosomal peptidases, these peptides come and get bound to the peptide binding groove of MHC2. And then, in presence of HLA-DM, the clip is removed. But if there is HLA-DO, this action is inhibited. So once the clip is removed, the peptide is bound, then this MHC2 along with the peptide is expressed out. And it talks to CD4 T cells. Yes, so we have understood what an exogenous presentation is and what an endogenous presentation is. But it is not so simple. We know, antigen presenting cells, it does not mean that it only presents to CD4 T cells. What if it is a viral antigen, right? It needs CD8 to come and kill it. Cell-mediated immunity has to come. CD4 means it will then proliferate, go talk to B cells, lot of time. So it needs CD8 also to come in. So what it does is, there is something called cross-presentation. So cross-presentation has got two major pathways. That's all we'll remember today. One is a vacuolar pathway and another is a cytosolic pathway. Through the vacuolar pathway, anything that is engulfed in the phagosome itself, it is degraded, and if MHC class 1 is there, it is then loaded onto the MHC class 1 and sent out. Whereas the cytosolic pathway is more complex. Cytosolic pathway, once these ingested amino acids or peptides come in, then it is processed in the phagosome and it exits into the cytosol. Once it exits into the cytosol, it starts behaving like an endogenous peptide, which is then loaded, which then goes through the proteasome, protease degradation, through the tap, it goes back to the endoplasmic reticulum. There MHC class 1 is waiting and all the same story unfolds, and then MHC class 1 can come out and present it outside. So here, even though the antigen is exogenous, there are mechanisms where antigen presenting cells can present it to CD8 cytotoxic T cells. So to summarize, what are the roles of MHC? One is to show self-peptides, especially in thymus, what Pankti has told us so beautifully. In the thymus, MHC actually shows self-peptides so that there is a lot of tolerance happening. Even overexpression of DO in the MHC class 2 pathway, if DO is overexpressed, what will happen? Clip is not removed. So there is no actual presentation of our antigens. So here, this also leads to tolerance to some extent. Then in infections, antigen presenting cells will take up, present it, and will induce immune response. Also in cancer, these cells will present cancer cells and will precipitate immune response and lead to cure. But then how can just these two molecules do all the work, right? So I think Sakir has told us about diversity and polymorphism. So what is polymorphism and what is mutation? A simple thing is if the change in the genetic sequence is in more than 1% of the population, it is considered as polymorphism. And if it is sporadic and one-off cases, then it is a mutation. So here what happens is one of these single nucleotides will get converted during the process of meiosis, and then this leads to a lot of alleles. So this has resulted in a number of polymorphisms in both HLA class 1 and class 2 alleles, and over the years the reported polymorphisms are ever increasing. So this variability that is there in both HLA class 1 and class 2, along with the diversity of the T cell receptor and B cell receptor, makes it possible for us to technically present infinite number of options to our immune cells. So very few applied aspects. Most of these things are already discussed. One specific thing that I want to discuss is absence of TAP protein that we discussed so much. It will lead to Bare lymphocyte syndrome, where there is skin necrosis, recurrent infections and other things. But other than this, we have already discussed about complement deficiency. Now complement is also part of MHC, that is class 3. So complement deficiencies, early complement deficiency will lead to autoimmunity, later on will lead to infections. But some MHC polymorphism can also lead to drug hypersensitivity. That is, they specifically present these drug molecules to CD8 cytotoxic T cells, especially class 1 And with this, most of this we are already aware of, allopurinol being HLA-B 58 and abacavir HLA-B 5701. So in predisposed populations, especially Southeast Asian population, the frequency of these alleles are higher, and it is recommended to screen for these polymorphisms before starting allopurinol. So I'll summarize quickly. HLA, it is a glycoprotein that is expressed on the cell surface. It is coded by MHC, which is at the 6th chromosome. And MHC or HLA, they are used interchangeably. They are of mainly 3 types, class 1, class 2 and class 3. And MHC1 we know presents endogenous peptides to CD8 cells. MHC2 presents exogenous peptides to CD4 T cells. There is also cross presentation happening with the help of MHC1, which presents exogenous peptides to CD8 T cells. And there's a lot of polymorphisms that are reported for several autoimmune diseases, immunodeficiencies, and in cancer. And there's a very important role in transplant, where if the MHC match is there at the allele level, then it is expected that the transplant uptake is good. Thank you.
Shankappa, great job.
Thank you.
What about this one thing that I've always never clearly understood is the shared epitope in RNA that also is related to the MHC class 2.
Okay. So shared epitope is a sequence in the MHC class 2, HLA-DR04 mainly. So, so basically what happens in autoimmunity is it is not one gene that is there. It is a haplotype that is there. Haplotype meaning usually a set of genes are inherited together. Yeah. So the shared epitope, what they say is if this person has a certain HLA-DR something 04 except 13, any of these, then they have a predisposition to present the QKRR, all these peptides to CD4 T cells and then proliferate RA. That is the concept.
If I can add to that,
Yeah.
if you read a textbook, you will see RA is associated with HLA, DR 0404, 0401, 0104. What is common between all these? What is common is that in the 11th position of that HLA, there is a specific amino acid, K. K stands for
Cysteine.
what? Okay.
Cysteine, no?
And then again 14th position is fixed. The 11th position and 14th position are fixed, and it is common between all these 0401, 01, 04, 04, 04, all of the DR. And overall, approximately 5 amino acids, what here mentioned are same, but more specifically at position 11 and position 14. Those two positions are very much fixed. And all the HLA which have this common epitope, because this is in the epitope of the MSAs. They are together called as shared epitope. So any HLA which has this 5 amino acid or approximately 4 of these in this fixed position from, may not be 5 for all, it may be even 4 from position 10 to 14. Those are combined together, and instead of calling them all the names, we give them one name, that is shared epitope.
Shankappa, can you just touch upon alleles? What do you mean by alleles? And
So it is a shared location in the gene which codes for the same thing, like for HLA-B27, the allele is 0405, that is the allele. So that's how they are named also. HLA-B27 becomes the allele, and after that it becomes the peptide. So the specific coding region in the chromosome